So now we're going to talk about tissues. There are four primary types. Epithelial tissue covers and lines. So think of covering and lining organs. Connective tissue provides support. So connective tissue providing support could be tendons in muscles. And then muscles enable movement. That's obvious. Nervous tissue controls work. Um, so you wouldn't be able to move your muscles if you didn't have the nervous tissue controlling that input to the muscle, so controlling the work it does. So the 6 examples of functions for epithelial tissue are protect, cover, and lines. So these are tissues that are protecting, covering, and lining other organs such as uh, the inside of intestines or the inside of thorax where they're covering the pleura and or lungs. Um, then they filter biochemical substances. This can even go down to your skin. Your skin does filter out substances from being allowed to be absorbed. They absorb nutrients, provide sensory input, again, your skin, and manufacture secretions and excretions. Characteristics of epithelia. They are organized into tightly packed groups to form sheets. There are apical and basal surfaces. The apical side of the cell is what faces the lumen or body cavity. When I think of lumen, I think of intestines. So the inner lumen of the intestine would be the inside of the intestine, and the apical surface of an epithelial cell would be facing that surface. The basal surface would be the opposite of that, and that would be what is facing the underlying connective tissue. So you can think of that as the apical surface is facing outwards, and the basal surface is what's actually connecting that cell to the underlying tissue. Lateral surfaces connecting neighbor cells, so just like lateral in the directional terms on either side, so right and left if there's a right and left. Cells are avascular, so just like terminology, which you should have gotten into this by now, avascular, anything with an A in front of it means non or not in basic terms, I guess. So avascular would mean lacking veins or vasculature. So that is a lack of blood supply. Just like anesthesia, a and an as a prefix are lacking. So aesthesia means feeling. So anesthesia or anesthesia means not feeling. So avascular is a lack of blood supply, and most cells are innervated. So again, thinking about your skin, if you were to prick your skin, you're going to feel that because it's innervated. I left this slide in mostly for the basement membrane because that is something that is brought up in your coursework to be able to discuss that term. But for tight junctions, desmosomes, gap junctions, I just left that in here as a slide, but I won't define those. So these are the types of cellular attachments, and the major types are tight junctions, desmosomes, gap junctions, and basement membrane. So basement membrane, also called the basal lamina, is the foundation of the cell. It's what cements the epithelial cells to the underlying connective tissue, and it is a non-living network of fibers. This helps the cell from being prevented being torn off, and is really what holds the cell to the layer underneath. One example given in your course notes is if in wound healing the basement membrane is damaged or removed in a wound or a deep cut, and it cannot repair itself, then the cell won't be able to regenerate. So that epithelial cell without the basement membrane is not going to be able to regenerate itself. So that is the important takeaway from basement membrane. So surface specialization of epithelial cells. There are different surfaces on these cells depending on where they are in the body. The smooth cells can be found in vessels so that they can allow for blood cells to pass easily. Microvilli are irregular shaped surfaces or finger-like projections that are found lining the intestines. If we think back to the parvo puppy where these microvilli are damaged and those finger-like projections are no longer capable of absorbing nutrients into the body, so the animal becomes sick from lack of nutrients, and this also creates diarrhea. So the brush border is the surface of the cell covered with microvilli, so it's just another name for what they are calling the collection of these microvilli. The brush border greatly increases the surface area of the cell, so the finger-like projections, because it is a long um, finger-shaped cell, there is much more surface area on all the sides of that projection, and this is what increases the absorption ability. Cilia. Cilia are thousands of tiny hairs. So these can be found in the lungs. So if we didn't have cilia in our lungs, then we would have a hard time getting rid of foreign invaders or small particles that can enter the lungs um, and which are brought out from mucus lining the cilia, and then you can cough it out. Um, this is a problem for Sphinx cats because they lack hair on their body on the outside, but they also lack hair on the inside. And because cilia are hair, a lot of truly bred Sphinx cats do not have cilia in their lungs, and it is really important for those cats to have dustless litter because they cannot get that dust and those dust particles out of their lungs because they lack the cilia. Simple squamous epithelium are delicate, thin, flat, and smooth cells that line the surfaces involved in passage of either gas or liquid, such as the gas passage in lungs where the oxygen is absorbed and carbon dioxide is released. And the cells have a mesothelium or endothelium capability. So simple squamous epithelial cells can be found in the chest cavity, so in the pleural space, the pericardial sac, which is around the heart, and the peritoneal cavity, which is the abdominal cavity. So these cavities are called mesothelium because they are inside a lining. So again, lining the pleural space, chest cavity, lining the peritoneal space in the abdomen, and the pericardial space around the heart. The endothelium is cells that are lining blood and lymphatic vessels. So they are inside those vessels, hence endo. When you think of simple columnar epithelium, if you are to think of these cells in relation to the gastrointestinal tract, that may help because these line the gastrointestinal tract from the stomach all the way to the rectum, and they are associated with absorption and secretion. So they are found in many excretory ducts as well as in the digestive tract. So the absorptive cells are going to be what are making up the gut lining so that they are able to absorb nutrients. And so this would be what is blanketed by dense microvilli to maximize the absorption and increasing the surface contact if the nutrient-filled lumen. The other cell is the goblet cell, and it is named that because of its wine glass shape. These cells are what manufacture and store lubricating mucus that is secreted into the luminal surfaces of the epithelia. So this would be probably the example of the mucus that can be found in the colon, and you can see animals that have diarrhea have mucus sometimes in that diarrhea that is lost. These goblet cells are responsible for the manufacture of this mucus and would be lining the luminal surfaces of the rectum and colon. Stratified squamous epithelium are multi-layered. They protect underlying tissues in areas where there are a lot of sloughing of this tissue or wearing off, so that there is continually going to be another layer right under that first layer if it is being sloughed off, so that the layer that is protecting is not exposed. So we find these cells in the mouth, the esophagus, and the vagina. And this is why your mouth will heal really quickly, because you have these multi-layered squamous epithelium in there, and they are continually being remade. Transitional epithelium are cells you're going to see when you do urinalysis because they line the bladder. So being able to identify these in a urine sample will come later, but there are transitional cells that will make up a lot of the cells that are found in urinalysis. They are a squamous cell, and it is a leak-proof membrane found in regions that require to expand and contract, such as the bladder. And they do have a specific look to them, and they are the cells that do get invaded by cancer. So a transitional cell carcinoma, or TCC, is something that you'll find in the bladder, and in cases where you are looking for TCC or transitional cell carcinoma cells, sometimes you'll do a traumatic bladder catheterization. So you'll on purpose put a larger catheter into the bladder and somewhat poke around in there, trying to slough off these tumor cells so that you're able to view them in your urinalysis. And they are quite easily to be identified when they are cancerous, because they will have more than one nucleus, and they come off in layers. So transitional cell epithelium are something that you'll see quite a lot of. So glands are a group of cells that manufacture and discharge a secretion, and they are classified by the presence or absence of ducts, number of cells that compose them, shape of secreting ducts, complexity of glandular structure, type of secretion produced, and the manner in which this secretion is stored or discharged. For the next couple slides, I won't go over every single slide because they are self-explanatory when you read them, and they really don't make up a giant portion of this class or the final exam. So just knowing the basics of the glands and exocrine, endocrine meanings and some examples would be what you should study for. So components of connective tissue, there are the ground substance, which is a medium through which the cells exchange nutrients and waste with the blood. They are amorphous, so amorphous means having no shape. Again, meaning without, morphous is just shape. Homogeneous, so homo meaning the same, so they are the same material without shape, such as glycoproteins. They envelop and protect delicate cells. They are effective obstacles for invading microorganisms, such as microbes. Extracellular fibers include collagenous fibers, which are strong, thick strands of protein collagen. They are organized into discrete bundles of long parallel fibrils, which in turn are composed of bundled microfibrils. Um, Basically, that just is how they are given their strength, because they have many fibrils in bundles, which are resistant to pulling forces. So collagenous fibers are found in tendons and ligaments and areas that are continually being pulled and stretched. Reticular fibers are thin, delicate, branched networks of collagen that still provide support even though they are not thick like tendons. They are providing support for highly cellular organs or endocrine glands, lymph nodes, spleen, bone marrow, and liver. And they are also found around nerves, blood vessels, muscle fibers, and capillaries. So instead of providing support by being thick and strong, they are thin and delicate and complicated, but still providing support for these delicate um, vessels, nerves, and fibers. Areolar tissue, I find, is a somewhat misleading name because it is from the Latin areola, which is supposed to mean small open space, but most of us females would not equate that to be that term. So when I think of areolar tissue, I think of breast tissue, which is incorrect. Instead, if you think of it being more of a packing supply or packing material that supports and cushions organs, then you may think of these air-filled bags that come in your um, packing supplies, I guess. Those are made to support and cushion whatever you have that's being delivered to you in a box. So that is one way to maybe think of the areolar tissue, is that it's connective tissue that is a small open space that cushions and supports organs and other delicate structures. So it is a good packing material. The pathological state of areolar tissue is edema or pitting edema. So during trauma or other pathologic states, the spaces in loose connective tissue can fill with excessive amounts of body fluid, which is called edema, and the connective tissue is said to be edematous. You can see this in swollen paws or a fractured bone where the surrounding tissues are going to become swollen and edematous. Pitting edema is, like I had said earlier in another video, where if there is so much fluid under the skin, if you were to put your finger into that fluid or into that tissue, your finger impression will be left in that skin area for a certain amount of time because it is pitting into the tissue. So that is pitting edema. There is so much fluid or swelling to a specific area that an impression is left after you poke that area. Adipose tissue is fat, and it is a loose connective tissue or areolar tissue that is just filled with fat cells or adipocytes. It is highly vascular. There are two main types, the white adipose and brown adipose, and they function as an energy storehouse where fat stores can be turned into energy. Thermal insulator, like that of a seal, they have a giant store of fat layer, which is an insulator in cold water, and it is a mechanical shock absorber. So lining the interior of your skin and your organs can be a shock absorber, although having too much of this obviously can be dangerous and inhibit the movement. The next couple slides are just describing dense regular connective tissue and dense irregular connective tissue. So dense regular are tendons, ligaments, and sheets of fascia, and they are relatively avascular, meaning again, lack blood supply. The dense irregular connective tissue are found in the dermis of the skin. Fibrous coverings of organs, such as the spleen, has a capsule, and other capsules such as joints. Elastic connective tissue is explained in the word elastic. They are found in body regions that require stretching, such as ligaments, walls of the arteries, stomach, bronchi, bladder, and heart. So these organs wouldn't be able to function properly if they didn't have the ability to stretch. Also found in spaces between vertebrae. Specialized connective tissues include cartilage, osseous connective tissue, which is bone, and blood, which is under the heading connective tissue. The cartilage and bone tissues are explained in the skeletal slides, so just read through them again and remember the words hyaline and the other components of fibrocartilaginous structures. Membranes are important in veterinary medicine, and in your notes, they want you to really dive into the clinical application on page 135 in chapter 5 as the key to a diagnosis. And this is just describing the different colors of mucous membranes and how we can look at these colors, which indicate different levels of disease or trauma. So icterus is the yellow appearance of a mucous membrane, also called jaundice, and this is usually involved with increased levels of bilirubin for reasons such as liver failure or hemolytic anemia, where the bilirubin is being expelled from the red blood cell. For hypoxia, you would see blue mucous membranes. So that is in an anoxic state. Again, an meaning without, oxic meaning oxygen, or hypoxic state, same thing. Hypo meaning below normal, and oxia meaning oxygen. Having a extremely red mucous membrane or hyperemia state, which is bright red mucous membranes, also called brick red. This can indicate a hyperperfused state, meaning hyper too much, perfused where blood flow is too much, I guess, in the mucous membranes. This can indicate a febrile condition or a hypertensive state. So febrile meaning that they are pyrexic, also a fever, and hypertensive meaning that they are their blood pressure is really increased. So we see both of these conditions in animals that have hyperthermia, being left in a car. They will have bright red mucous membranes with a CRT, or capillary refill time, of less than 1 second. And the last mucous membrane color that we look for is white, and this indicates the loss of blood or shock. So if they have white gums, then they either have had an incredible amount of trauma or blood loss at one time, and or they are in shock. So all of that blood has been shunted away from the mucous membranes and to areas it is needed most. If we think of the sympathetic nervous system and the fight or flight again. Mucous membranes in plural is mucosae, and you'll find mucous membranes lining the surface of any area where large quantities of mucus is needed. Mucus is a collection of water, electrolytes, and the protein mucin. Some mucosae can also absorb and transfer nutrients, such as that found in the epithelial layer in the intestines, as well as secrete mucus. Serous membranes, or plural serosae, they line the walls and cover the organs that fill closed body cavities, such as the chest cavity or abdominal cavity, as well as pelvic cavities. So in this picture here, there is the parietal peritoneum and the visceral peritoneum. The parietal layer is what the cavity wall is lined with, and the visceral layer is what is covering the outside surfaces of the organs. So you should know those two words, parietal again being the layer covering the peritoneum or the peritoneal space, and the visceral layer is the outside covering of the organs. Serous membranes also produce watery serosal fluid, which is a transidate. So a transidate is a fluid that does not contain mucin, so it is not mucousy, but it does contain electrolytes in a thin, watery solution. So there is also pleural fluid in the thorax, peritoneal fluid in the abdomen, and pericardial fluid in around the heart. And transidates are usually what is found in these areas. Expanding more on serosal fluid, there can be small amounts normally found in body cavities. Excessive fluid production, though, is pathologic or can indicate a pathologic condition such as liver disease, which would produce the effusion ascites. Nephrosis would be the fluid produced from kidney failure. Peritonitis being the fluid that would be produced in a inflammatory state of the peritoneum. Adhesions are something else that you should know about. They are serous membranes that are damaged, so the production of the serous fluid may be impeded, and abnormal connections, which are called adhesions, may form between the parietal and visceral layers of organs. They can also happen between the visceral layers of an organ to another organ, and this basically is connecting these organs or other contents of the abdomen to the wall of the abdomen or to each other, and can be found in animals that have had several foreign body surgeries. They start having adhesions from their intestines to other places in the parietal cavity, and when it is connecting another intestine to another one, then sometimes the movement of food and other substances through the intestines is impeded because the intestines now have right angles and other strictures and closed off areas from these adhesions. For muscle tissue, I will just go into the three types and you can look more in detail in the next three slides. But the big takeaway from these muscle types is that skeletal is muscle that is attached to the skeleton, and it is voluntary muscle, so that is something that you control when you run or kick a ball. Smooth muscle is mostly involuntary, so it lines the hollow organs such as blood vessels, the uterus, intestines, and stomach. And it is something that you cannot consciously control. And then cardiac is only found in the heart, and it is specialized for pacemaker cells within the heart muscle. So only found in the heart and is another involuntary form of muscle. Inflammation, the first step in tissue healing, and tissue healing and repair is something you should definitely know. So the first step to response of injury or disease processes is vasoconstriction, so the constriction of blood vessels, and later vasodilation, so the opening up of blood vessels, edema or swelling, clot formation, phagocytosis by the white blood cells, and release of histamine and heparin. The steps in the process are vasoconstriction, which is small blood vessels constrict to stop the hemorrhage, and later vasodilation, which helps when histamine and heparin molecules are released, and this allows the blood vessel to dilate. This dilation increases blood flow to the area, which is also the reason that heat and redness is seen. This also brings oxygen and nutrients to the site. Clots stop the bleeding and isolate the wound to help prevent bacteria and toxins from spreading to other areas or tissues in the body. Phagocytic cells such as neutrophils and macrophages assist in removing debris and invaders by engulfing them, and they only do this for a few hours until they die. So pus is accumulation of these dead and degenerated neutrophils and macrophages that have collected in that injured area. When the blood flow returns, then the capillaries return to the normal size and fluid leakage into the affected area can stop. Swelling, heat, and redness then begin to subside. The regeneration or fibrosis of the area then begins. So epithelialization covers granulation tissue. Eventually, the scab is pushed off, and you are left with a fibrous scar. The classifications of wound healing are first intention, where the wound edges are in close apposition. Apposition meaning like how the edges are brought together or opposed. So first intention healing can be a very superficial wound where the apposition of the edges is already in close proximation with each other, or it can be that it is a deeper wound where the apposition of those two wound edges has been brought in close proximity because they are held together with staples or sutures. No granulation tissue is formed because there is no area for that to need to be produced because the wound has been brought together. Second intention is when the wound edges are not close in apposition or closely opposed, and there are separation between the two edges. And this is when granulation tissue needs to be formed to fill in that space or gap, and that's when scarring results.